Ladies and gentlemen, distinguished speakers and participants from around the world, good morning, good afternoon, and good evening. I am Jang Homin, Chief Director of the Korea Institute for Promoting Asia Biosafety Cooperation. We call KIPABiC in brief. It's my great pleasure to welcome you all to today's informational webinar on synthetic biology, technological developments, and policy discussions. KIPABiC is hosting this webinar in response to numerous requests and feedback from individuals eager to gain deeper insights into the rapidly advancing field of synthetic biology. As a multidisciplinary domain, synthetic biology has attracted attention across various sectors due to its potential to address some of the world's most pressing issues, including biodiversity loss, climate change, conservation, hunger, and vector-borne diseases. We are thrilled to bring together a diverse audience from government, academia, industry, NGOs, and civil society. With over 900,000 participants, the number is registered number. Well, with them expected to join us from more than 50 countries worldwide, this webinar highlights the strong global interest in understanding the latest developments and opportunities in synthetic biology. This webinar aims to introduce the essentials of synthetic biology, including the latest technological developments and key policy discussions currently shaping this rapidly evolving field. And we hope this session will not only inform, but also prepare you for the upcoming discussions at COP-MOP 16 in Cali, Colombia. Today's event is organized by KIPABiC in collaboration with UNEP and 4 participating countries: Bangladesh, India, Mongolia, and the Philippines. It's part of the capacity building and knowledge sharing initiative and the GEF multi-country project titled Promoting the Safe Application of Biotechnology through Multi-Country Cooperation in the Implementation of National Biosafety Frameworks in Asia. It is a multi-country 3-year project involving 4 countries that I just mentioned, with GEF funding of $1.09 million and co-finance from the 4 countries and the government of the Republic of Korea. We have UNEP for technical supervision, while KIPABiC is involved as the lead executing agency and project management unit. If you are interested in more, please visit the link that you would see on the screen. To share a little more about KIPABiC, we are a non-profit organization based in South Korea, deeply committed supporting countries, mostly in Asia, through capacity building initiatives and collaborative approaches in biosafety and biotechnology. This is the agenda for the webinar. Our webinar will be divided into 2 sessions. One is on technological developments and another policy discussions. To begin our first session on technological developments, it's my pleasure to turn this microphone to Dr. Weber to moderate the session. Dr. Weber, I would like to, yeah.
Thank you so much for the kind introduction to the objectives of this webinar And since we have a very short time, 1.5 hours only, and a very important topic of discussion today, we will move on to the session 1, which is on technological developments. So as Dr. Jang said, that synthetic biology is an area of significant interest to all, and at the same time, the area is under debate under the various international conventions, particularly the Convention on Biological Diversity that we are focusing today. So we have 2 presentations in this session from very learned experts. So the first one on the introduction to synthetic biology and the latest advances, and the second on the developments under the Convention on Biological Diversity on synthetic biology. So I have my great pleasure to introduce the first speaker, Dr. Felix Moronta Barrios. He is from the Regulatory Science Group at International Center for Genetic Engineering and Biotechnology, Italy. Dr. Felix has been specializing in bridging the biotechnology and sustainable food security and extensively trained on this interface between the science, policy, and society. The most relevant achievement of his in the context of today's webinar is that he has co-authored the CBD Technical Series 100 on synthetic biology and also has contributed to drafting of various other documents like voluntary guidance in the case-by-case risk assessment of LMOs containing engineered gene drives. So I will request Dr. Felix to kindly give his presentation. Thank you.
Thank you very much, Viva, for your kind presentation. And I'm grateful to be here with all of you today, and also for the kind invitation of KIPABiC for this opportunity. I'm going to proceed to share my screen because I have prepared some slides with some key messages that are important for the topic of today. Okay, so I will introduce synthetic biology as a field and will shortly comment about the latest advancement in this field. And the content of my short talk is the points that you can see here. I will try to address what is synthetic biology, the difference between synthetic biology and genetic engineering, the technologies or tools and research areas supporting synthetic biology, application and products, and also those applications with potential implication to biodiversity. And I will close my talk talking about just one particular example of the latest development, that it is engineered gene drive. To start, let's start by answering or trying to answer, what is synthetic biology? And well, this is a little bit tricky because there is not an agreed international definition of what is synthetic biology. We can then consider the definition that is used by the Convention on Biological Diversity that you can see here. And it reads that synthetic biology is a further development and a new dimension of modern biotechnology that combines science, technology, and engineering to facilitate and accelerate the understanding, design, redesign, manufacture, modification of genetic material, living organism, or biological systems. And the term synthetic biology was first used in the early 20th century, but the field as we know it began to take place in the early 2000s. And a significant milestone include the synthesis of the Mycoplasma genitalium genome, and also the creation of the first self-replicating synthetic bacterial cell, and then the discovery and manipulation of CRISPR-Cas9 technology further revolutionized the field. And it is crucial also to acknowledge and to mention that the decrease in synthetic DNA cost and sequencing have accelerated innovation, making synthetic biology more accessible and powerful than ever. And this historical milestone highlights the rapid advancement in synthetic biology in the last decade. One recurrent question that I also have had myself is, what are the differences between synthetic biology and genetic engineering? First, we need to recognize that both fields are using biotechnology to improve or create new biological products by manipulating DNA to achieve specific outcomes. This is the main similarity between the two fields. But we can, for sure, identify some key differences regarding scope, approach, complexity, end goals, standardization, and some techniques that are different between they both. Regarding the scope, genetic engineering typically focuses on modifying the genes within an existing organism, while synthetic biology aims to create a new biological system or redesign existing one, often from scratch. The approach, genetic engineering, modifies intrinsic, the natural, let's say, genetic material, while synthetic biology use principles from engineering to design or build new genetic construct. And the complexity. Genetic engineering involves modification typically at the gene level, while synthetic biology can involve creating new metabolic pathway or even create whole synthetic new genomes. Regarding the end goals, genetic engineering mainly for improving or adding specific traits, and synthetic biology to construct, again, new biological system with novel functions. And one key point is the standardization. In genetic engineering, there is less emphasis on using standardized parts, while in synthetic biology, there is a strong emphasis in using a standardized, reusable biological part. One example are biobricks. And just to bring some example of the techniques here, this is the classical gene cloning or knockout, CRISPR, transgenic organism. And in synthetic biology, gene synthesis, chemical gene synthesis, modular genetic component pathway engineering, or even whole genome synthesis. And what about the supporting technologies, tools, and areas of research? Synthetic biology, or SynBio, relies on a suite of supporting technologies and tools that enable the engineering and creation of biological components. This tool could include, again, DNA synthesis, directed evolution, genome editing, engineered gene drive, RNA interference, artificial intelligence, machine learning, biobricks, biofoundries. I'm not going to go deep into each of those, just mention them. And also synthetic biology covers several areas of research, such as genetic circuits, engineering of protein metabolism, genomes, protocells, xenobiology, or even cell-free systems. And these create a complex and rich scenario for developing new applications and products. And this application span across various fields. For instance, in medicine, revolutionizing the disease treatment and prevention. In agriculture, creating pest-resistant and stress-tolerant crops. For instance, environment, enhancing bioremediation and sustainable manufacturing, or in energy, production of biofuel that are more efficient. And this application, general application, illustrate the transformative potential of synthetic biology across different sectors. Now I will present some specific synthetic biology applications and products, which are categorized according their intended release setting, if they are released or used in contained setting, in managed setting as an agricultural field, or if they are intended to be released in the wild. And at the same time, if they are commercially available in advanced development, or they are under research. And now I will go through these categories in the following way. Here you will find the intended released environment, and with this mountain or pyramid, the level of development, if they are in research, advanced development, or commercially available. This is not an exhaustive list, it's just some examples. Under research, for being used in contained settings, well, there are, for instance, development of minimal cells, non-native nucleotides and amino acids, recreation of extinct viruses from chemically synthesized DNA, and other examples. In advanced development, the biofabricated wildlife products, or culture-related products, or plant-based vaccines, or even engineered probiotics for in vivo delivery of medicines. And there are most of the SynBio products that are commercially available belong to this category that are using contained settings. And they are biopharma products, fabric products, cosmetic and fragrances, food and food ingredients. The next category of products are those that are intended to be used or released in managed environment, like agricultural settings. Under research, there are a lot of products there, typically genetically engineered bacteria to be used in agriculture. Something more complex like insect delivery of modified viruses, to modify the crops. Project for the de-extinction of extinct animals, or even the transient modification of agricultural plants using RNAi spray. In advanced development, we can find genome-edited crops on farm animals, or even engineered gene drive in mosquito and pest, and also self-limiting insect. And commercially available, there are already a couple of examples. One is the genome-edited crops, soybean and oilseed rape, and also a biological nitrogen fertilizer that is based on engineered bacteria using SynBio approach. The third category, in the wild, there are under research products, SynBio products to be released in the environment. These are genetically engineered bacteria for environmental applications such as bioremediation, biodegradation, or biomining, and also for conservation purposes or improving the resilience of wild population. We have not categorized products in advanced development or commercially available, but they are coming. And I want to go a little bit deep in these two examples. These are like synthetic biology application with impacts on biodiversity. And this application, we can divide it in two: those that are intended for conservation benefits and those that are not intended for conservation benefits. In the first category, we have, for instance, application to reduce threat, or the mitigation of biological threat. For example, to eradicate invasive rodents that cause extinction in other species on island. This could be done through engineered gene drives, or increase resistant adaptation, or the resilience of organism. For instance, modifying the genes of corals to increase resistance toward warming ocean. This can be done. It is under research by genome editing. Instead, those applications that are not primarily intended for conservation benefit, but they could affect biodiversity, Are those for pest control, for product replacement, agriculture, or environmental engineering? And now I want to comment on one particular example of technology that is engineered gene drive that can be used for intended conservation benefit or not intended, but still have impacts on the environment. But what is an engineered gene drive? Well, the advancement in synthetic biology, in CRISPR, have delivered the molecular tools. This is from the first report was a few years ago, let's say, to enable the design and development of a wide range of engineered gene drive. And an engineered gene drive spread genetic modification at a rate that is greater than expected by the normal, let's say, expected Mendelian inheritance. And this characteristic enabled them to proliferate rapidly within sexually reproducing organisms. And this is a schematic example of the difference between the inheritance of gene drive organism. Well, eventually, the entire population will have that genetic modification of sexually reproducing organisms. And unlike other GMOs, the engineered gene drive are often designed or constructed to be applied in wild organisms. And they are also designed to be to disperse beyond their initial release location and persist in the environment, in target population over extended period of times and generation. And there are two strategies for population control using engineered gene drive. The population replacement, you can see a schematic example here, where the modified organism eventually take over the wild population, or population repression, where the introduction of the modified organism produce the collapse of the total population of the target organism. And as a result, the ecological impacts of engineered gene drive have the potential to be far-reaching. And while this technology, gene drive, are well developed in yeast and some animals, applying this technology in plants, in crops, has been a major technical challenge. However, this is changing very fast. Now, genetic intervention in wild plant population using gene drive is possible, according to the latest research and models. Actually, in June, 3 months ago, two groundbreaking studies were published that successfully introduced gene drive element into Arabidopsis. And there are a screen capture of the two here. And well, this makes us think, and this is, oh, I'm hearing a sound, sorry. And I am asking you as a reflective question, how is the regulatory landscape evolving to accommodate such transformative technologies, or should evolve to accommodate it? And before closing, I wanted to highlight this CBD technical series on synthetic biology that Viva already commented to you. I am one of the co-authors of this comprehensive report covering all these areas that you can see here in this list, and you can go on the CBD webpage and download it for free. And well, my take-home messages are that synthetic biology is a rapidly growing field with transformative potential across multiple sectors. And while synthetic biology and genetic engineering overlap, SynBio uniquely applies engineered principles to design new genetic construct pathway or even entire synthetic genomes. Their products can directly or indirectly impact natural environments and biodiversity, and engineered gene drive could significantly alter ecosystem with both positive and negative effect. And again, I want to repeat the reflective question. how is the regulatory landscape evolving to accommodate these transformative technologies? And in closing, I wanted to present you my institute, International Center for Genetic Engineering and Biotechnology. We are an intergovernmental organization originally established under the UN system. And you can see in this map the member states that belong to our organization. And I am part of the regulatory science group. And in my group, we help national and international agencies in regulating the biotech product. I will be happy to discuss this later in other conversation. That's all, and thank you so much for your attention.
Thank you, Felix. Thank you so much for an excellent overview in such a short time. 15 minutes is too short for explaining the details of the technology, but you did an excellent job. Thank you so much. We'll come back. There are some questions for you, but we'll first hear Felicity and then come back to the questions. I hope you'll be with us. So, Dr. Felicity, we have the next presentation from Dr. Felicity Keiper. She is a senior expert in global regulatory policy in the seeds and traits business of BASF. She has worked in agriculture biotechnology for more than 25 years, and significant part being in the regulatory affairs. Particularly here in the context of synthetic biology and CBD, she has been a member of the ad hoc technical expert groups which are set up by the CBD from time to time for the intense discussions, detailed discussions on these topics of new topics. And Dr. Felicity has been a member of 2 of such groups, and since almost 10 years working on this area. And it's very kind of her to have agreed. It's very late, I think, late time in the Australia. And thank you, Felicity, for making yourself available at this time. And we look forward to hearing your presentation. Over to you.
Thank you, Viva, and to KIPABiC for the opportunity to participate in this webinar. And my slides, Tashi was going to show my slides. But yeah, thank you for
sharing the slides.
Thanks. Yeah, so yeah, thank you for the opportunity to talk to you today about synthetic biology developments under the Convention on Biological Diversity. Next slide, please. And I will begin with, thanks. And I will begin with a brief overview of the convention. So the objectives, so this convention is the primary international forum that's considering developments in synthetic biology in the context of managing potential environmental impacts, because the convention is a broad environmental treaty, but it does have specific provisions for the regulation of biotechnology. And these are connected to those first 2 aims of conservation and sustainable use. And the convention also has provisions for access to genetic resources and the fair and equitable sharing of the resulting benefits in support of that 3rd objective of the convention. And that objective is also relevant to synthetic biology. And this was a trigger for work that is now happening on digital sequence information under the convention. I won't be going into that topic, but just mention that that is also a substantial area of work under the convention. Next slide, please. And the specific regulatory provisions in the convention are found in Article 8G. So this requires parties to regulate the environmental risks associated with the release of living modified organisms, or LMOs, resulting from biotechnology. And the convention also has a broad definition of biotechnology, which is reproduced on the slide. This is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific uses. So I'm taking the time to read that because it is broad, any technological application. If you are I'm often asked about definitions, and if you are looking up definitions of synthetic biology, or you try to draft one yourself, as I have, you'll likely come up with something that is very consistent with this or fits within it. It's broad enough to include different approaches, like Felix has just talked about, different types of technologies and different types of resulting organisms. And now the convention also provides the basis for a protocol that sets out regulatory procedures. So this led to the development of the Cartagena Protocol on biosafety that entered into force 10 years after the convention. And the cornerstone of that protocol is its risk assessment framework. So that includes fundamental principles of risk assessment, general methodology. It also sets out processes for decision making. And these are reflected in many national regulatory frameworks for environmental releases of LMOs. So next slide, please. So how does synthetic biology fit in here? In 2011, we had the first submissions for synthetic biology to be considered as a new and emerging issue relating to the conservation and sustainable use of biodiversity. So the convention has an established process for this. It requires submissions to be made that address a list of criteria. These are set out in the COP decision that's highlighted on the slide. And the subsidiary body on scientific, technical and technological advice to the convention, known as SBSTA, that body reviews these submissions and it makes recommendations to the COP. So these submissions were triggered by some very high profile publications at the time. And Felix has just shown a couple of these in his presentation that reported the chemical synthesis of microbial genomes. And these really caught the attention of the public. There were widespread media about them, also caught the attention of policymakers. There was a bit of a hype phase, a lot of hype around the expanding possibilities with biotechnology, and also discussion around the scope of existing regulatory mechanisms. So the SBSTAs and the COPs that have considered this question, they haven't been able to reach a conclusion on whether or not synthetic biology is a new and emerging issue. At COP12, we had the establishment of the first program of work on synthetic biology. So this was sort of separate. This is starting to sort of separate from the issues of new and emerging, that agenda item. It was a subtopic of that. It's beginning to emerge as a topic of its own here. And this has been renewed at every COP since. So that was 2014. So we've now had a decade of dedicated work on this topic under the convention. And this work has been supported by ad hoc technical expert groups, which I'll call RTEGs, and I have served on those as an industry representative. Next slide, please. So from COP13, we saw synthetic biology become a fixture on the CBD, on the COP agenda. Also at COP13, we saw a new program of work. This came out from under synthetic biology. This was established for digital sequence information, and that continues today. And then more recently at COP15, which was the last COP in 2022, the synthetic biology program of work changed in substance, in that up to that point, it was the programs of work were tasked with deliberating lists of questions about synthetic biology, but here it changed to the specific task of horizon scanning. And we also saw the establishment of a new program of work on risk assessment under the Cartagena Protocol. So this was on the synthetic biology topic. It's a major topic under discussion in that work on engineered gene drives. And this also involved an RTEG that I served on as an industry representative. So for COP16, which I've highlighted there for the convention, that will make a decision about the outcomes of the horizon scanning work. And also COP-MOP 11 for the Cartagena Protocol, which will be held at the same time. So both of these meetings are coming up in October, so very soon. So COP-MOP 11 will be making a decision about the risk assessment program of work. And for both of these topics, there was a recent SBSTA meeting. This was SBSTA number 26, so they're numbered by how many meetings they've had. That was in May this year, and that reviewed the work of those two RTEGs, and it produced recommendations for the decisions for parties to be making at those meetings in October. Next slide, please. And this is a very brief summary of a decade of work here. Since 2011, the intersessional program, or more than a decade, these intersessional programs of work on synthetic biology have largely focused on this series of questions about it. And, as mentioned already, the new and emerging issues question, and this remains unresolved. It was considered by SBSTAs. It's also been considered by one of the RTEGs. There is a wide divergence of views on this, from it not being a new and emerging issue that requires ongoing programs of work, to it being something that it does require continual monitoring and assessment, irrespective of whether or not it is a new and emerging issue. Also, an operational definition for synthetic biology, as Felix has just shown. One was developed by an RTEG, but it wasn't officially adopted by parties. So this question remains unresolved at the international level for similar reasons: divergent views on what it is, and on the need for it in this forum, and the potential positive and negative implications of synthetic biology for the convention have been considered at length. And the RTEG reports, they generally broadly list developments in technologies and potential applications and potential impacts. Areas that are particularly informative, at least for developers, so I work for a developer, the considerations of the ongoing applicability of existing regulatory instruments for the organisms developed. And then at the last point there, we have the current area of work on horizon scanning, monitoring and assessment. Next slide, please. So as we know, the outcomes do not include an agreed definition, and this was a task for that first RTEG. And this was and remains a contentious topic. It comes up repeatedly at the meetings. There are parties that do want this resolved, and there are others that don't see the need for this, because there is general agreement that synthetic biology is within the scope of that broad definition of biotechnology that's in the convention. And this means it is within the scope of Article 8G that I mentioned earlier, that has regulatory requirements. Also, that most organisms developed or currently in development remain within the scope of LMO, as defined by the Cartagena Protocol. So this means that the existing regulatory provisions of the protocol remain applicable. So this is why some parties, they don't see the need for ongoing programs of work on this topic. It's within the scope of the instruments we already have. And importantly, the applicability of the protocol includes its risk assessment framework. So I've mentioned that there is a program of work for engineered gene drives, and this is for the development of additional voluntary guidance materials to support case-by-case risk assessment of LMOs containing engineered gene drives. So these are a type of LMO that is within the scope of the protocol. Next slide, please. So moving on to developments in the current intersessional period, and this is the period between COP15, the last COP in 2022, and the upcoming COP16 in October this year. And this is where we saw the synthetic biology program of work shift from sort of those lists of questions to this development of a horizon scanning, monitoring and assessment process. So the RTEG worked to define that process, and it applied an abbreviated version of that in one cycle of horizon scanning. The last COP also stated that there would be no more debate about whether or not synthetic biology is a new and emerging issue. So it was agreed to leave that question unresolved with work on the topic to proceed. Next slide, please. So the work completed since COP15 was considered, as I mentioned, by the recent Substar, and it produced recommendations for the future work on this topic. And I will be brief here because I think the next speaker, Wadzi, will go into this in more detail. But in summary, the recommendation has 3 substantive areas, including a section on capacity building and development with a new proposal for the development of an action plan. A second area is what to do about the horizon scanning work, continue it or not, what about the process, what do we do with the RTEG? And the third area is the terms of reference for an RTEG should it continue. So these are tasks assigned to it for the next intersessional period. And a lot of this text is bracketed, so that means it was contentious at Substar, it wasn't resolved, and this will have to be negotiated by COP16 at the meetings in October. There's a lot of brackets here, so there are several possible scenarios that could result. And there's not really a clear view on what parties want this program of work to deliver. You know, the possibilities are a mix of things like returning to some of the outstanding issues I've mentioned, the definition, the new and emerging issues analysis, continuing or not the horizon scanning process in some form, providing advice that contributes to that capacity building action plan, and also in the identification of benefits. And I have highlighted the word benefits for a reason. Next slide, please. And as a final point, I wanted to mention this sort of overarching implementation priority for parties to the convention. And this is the Biodiversity Plan, also known as the Kunming Montreal Global Biodiversity Framework. So this includes a biosafety target, target 17, that includes the reference to Article 8G that I mentioned earlier. Also, the handling of biotechnology is set out in Article 19, and that is what is the basis for the Cartagena Protocol that sets out procedures for that. But Article 19 also talks about benefits of biotechnology. It talks about the results of biotechnology, but it also talks about effective participation in biotechnology. And that's where this capacity building plan that's proposed in the synthetic biology work is, heading towards. So as a final comment is to share my opinion that parties, when they're considering what they want the synthetic biology program of work to deliver, when they're deciding on the structure of this at COP16, that they consider the capacity development needed in a broad sense to support their implementation of this target, as well as the realization of benefits in the context of implementing other targets in the biodiversity plan that biotechnology has the potential to contribute to. Next slide, please. And this is my final slide. And with that, I'll finish. So thank you for your attention.
Thank you, Felicity. Once again, it's really a challenge to present decades of work in 15 minutes.
Yes.
But thank you for giving such a summary, presentation in a very nice way. So I think we are a little behind, but we will take 1 or 2 questions before moving on to the next session. Here, if I look at these questions, I think you also can see in the chat box. Basically, once again, we are revolving around what is the definition of synthetic biology, what is included and what is not included, and what is your suggested definition. The second major issue which is coming up is about the regulatory concerns, whether it fits into Cartagena Protocol and so on, and how does it diverge from the regulations. So when I was looking at this, I was just thinking that this is the issues which are being, discussed in COP, and the similar issues are coming up here. So maybe I will request Dr. Felix first to kindly give his remarks on basically the definition, what is included, not included, And also there is a question on how these products, can be used to control loss of biodiversity and sustainable use of plant wealth. You did touch upon it, but maybe you can once again, explain that.
Yeah, thank you so much, Viva. And Felicity, I really enjoyed your presentation. Thank you. You too. Okay, definition. I mean, as Felicity said, it is a very contentious topic. It is heavily bracketed in the discussion and decision of the SATA recently. So the definition that many, or not only national but also international organizations are taking is the definition from the CBT. It is a operational definition. It is very broad, and because it is broad, it is also considering many technologies. So, and then every jurisdiction, every country, every regulatory agency can interpret the definition under their own perspective. And this means that what it is seen bioproduct for a regulatory agency, it is not a seen bioproduct for a different one. This is because, again, the operational definition is very broad. And there is not that correct and incorrect definition or consideration of seen bioproducts. This is something that it will depend on every jurisdiction. What I presented in my talk is something more technical about the key difference between genetic engineering and synthetic biology that could help in the discussion. But again, it is not written in stone. And again, every regulatory agency has, using the operational definition, has their own perspective on what include and what cannot be included there. The other question was about the uses, isn't it?
Yes. Use particularly on the conservation of biodiversity. Controlling loss of biodiversity and sustainable use of plant wealth.
Okay. How can be used to benefit biodiversity? Yes. Yeah. Okay. For instance, it already exists in lab. In lab setting, it has been already developed. Engineer gene-drive mosquitoes to fight the avian malaria, not the human, but the avian. I mean, there is a huge problem with this parasite that is infecting some bird population in Hawaii. In the middle of the Pacific, and the bird population are decreasing in a very concerning levels. And one of the approach, instead of applying insecticides all over the island, is to try to target that particular species of mosquito with gene-drive mosquitoes to fight the avian malaria, and those try to rescue and conserve the population of bird in that island. The same in the Galapagos Island, they are thinking on how to release engineered gene-drive mice or rats to fight the invasive alien species of rats that are decreasing the population of lizard and turtles in Galapagos Island. So there are many examples that are already under research and providing computational models of changes of population, etc., on how the gene-drive animals could help in the conservation efforts. And more or less the same with in agriculture, in plants, not specifically with gene-drive, but with genome editing. Try to improve the resilience, the climate resilience of crops, or the pest resistance of crops using genome editing approach. That I see in one of the questions, that if this gene edited crop is considered as seen bioproduct or not. Again, it will depend. It will depend of the jurisdiction. And for one, I don't know, readers of the CBT technical series, can be, yes, a symbiotic product, but for other don't. And there is not again, there is no an incorrect question there. It would depend on the interpretation of each jurisdiction.
Thank you, Felix. And this is the definition is going to be the key to everything. And now we also heard from Felix that from Felicity that the biotechnology definition itself covers everything. So maybe that's a better way of dealing with it. There's another question addressed to you, but there are similar questions regarding the guidelines or policy documents by the CBD. And I think since Felicity has been a member of the both ACTEC on synthetic biology and also RARM, maybe I will ask her to give her insights on the status of how the issue is being dealt with respect to the policies and regulations under the protocol. Over to you.
Okay, so as I talked about in my presentation, some of the early work by the programs of work, early programs of work with the RTEGs, these were questions that they were looking at. So the applicability of the existing regulatory framework, where there might be gaps, and conclusions there were that most LMOs that are currently in development or have been developed remain within the definition of most organisms remain within the definition of LMOs in the Cartagena Protocol. So these are within regulatory scope of the Cartagena Protocol and national implementation of that, depending on how those national frameworks are defining LMOs. Also, other national frameworks that countries may not be parties to the Cartagena Protocol, but generally the definitions are quite consistent. On a risk assessment, so there's been some, I mentioned the program of work at the moment under the risk assessment under the Cartagena Protocol, and this has been to develop additional voluntary guidance materials to support risk assessment of a specific type of LMO that is considered to be a product of synthetic biology. So this is containing engineered gene drives. So it's important to note there that those guidelines or that guidance wasn't considered necessary due to any gaps in that regulatory framework, in the risk assessment framework in the Cartagena Protocol. It was, but it was considered that it may be useful in that these are different types of LMOs compared to what we have the most regulatory experience with. Generally, we're dealing with LMOs that are, for example, I work for a biotech crop developer. These are released in highly managed environments. They're not designed to spread beyond, far beyond the release sites. So LMOs containing engineered gene drives, they're developed for a different purpose, and there may be different types of questions that need to be asked to properly inform a risk assessment. It's not that the risk assessment itself methodology needs to be changed, or that the fundamental principles change. So the outcome there is a guidance document that will be considered by the parties at COP-MOP 11. In my opinion, I hope that they decide to adopt it because this is a way of it explains a way of doing a risk assessment that is based on a problem formulation approach. So that isn't specifically set out in the Cartagena Protocol. It's an approach to doing risk assessment that is focused on testing with scientific evidence plausible pathways to harm. And this is an approach that can be used irrespective of the technology used, whether it's new technology, older technology. New technologies are generally grouped into these synthetic biology discussions. And it doesn't matter what type of LMO. This is a broadly applicable approach. So this is a new resource that's been developed under the protocol to support risk assessment in a way that's it's like an emerging best practice. So as I've said in my presentation, I think that as there's been as this it's been fortunate that this program has been spread out over a period of time, so it's enabled quite thorough consideration of these conclusions that, you know, what's being developed still is within the existing regulatory framework, and where there is specific questions where that need some more attention, for example, in doing a risk assessment for engineered gene drives, that we can develop those resources.
Thank you. Thank you so much for clarifying. And of course, I think KIPABiC is also planning to have another informational webinar on the topic of gene drives. So we will hear more about that regulations. I think our time for the session is over, and I would like to thank the KIPABiC for giving us this opportunity to presentations and be moderating this session. And over to you, Tashi, for the next session.
Thank you, Dr. Viva, for moderating the first session. Yeah. And yeah, here I have to say that I'm sorry for missing out the introduction of you just before I turned micro over to you. I'm sorry for that. Well, in a way, you may not need, detailed introduction of you because you are already well known, so much well known globally as not to need any detailed introduction of you. Nevertheless, I'd like to give some brief introduction of you for the people who may know more about you. Dr. Viva is the Chief General Manager of Biotech Consortium India Ltd, with over 25 years of experience in biosafety and regulatory aspects of GMOs. She is a leading expert in the Indian biosafety regulatory framework. Dr. Viva has played a significant role in policy advocacy, capacity building, and the development of guidelines, making substantial contributions to biosafety initiatives across India and South Asia. Thank you once again, Dr. Viva, for wonderfully moderating the session. Okay, now we move into our second session, which will focus on policy discussions. My pleasure to introduce our moderator, Dr. Alex Usbini. He is the UNEP-GEF Portfolio Manager for Biosafety, with more than 2 decades of experience in biotechnology, biosafety, and policy advocacy. He oversees the development and implementation of National Biosafety Frameworks globally, bringing extensive expertise in molecular biology, regulatory science, and project management. Dr. Alex, we look forward to your expertise and guidance as you lead us through session 2. Dr. Alex?
Thank you, Dr. Jang. Thanks to the last session and the presenters, we're going to go into a second session on policy. I'm happy the framers did not make it policy and regulatory issues, because the regulatory issues are still in discussion to provide global guidance. So that is one of the reasons. The issues that are going on now includes where does synthetic biology sit within the CBD and its protocols? Is it purely on the convention? Is it an ABS issue? Is it a biosafety issue? Aspects of it has already been handled or is going on under risk assessment. So we would, in this session, have 2 key areas. One is on the global issues and preparation towards the COP. And then there'll be 2 presentations from the Asia region. We were hoping to get one from Africa, but because of time, we could not get a presenter. I would attempt to introduce all the three speakers first, and then call on the first speaker to go ahead. For Dr. Weber, I will not introduce again, so I'll only introduce the two with all protocols observed and the necessary permissions given to me. I'll go ahead that way. So the first speaker under this session is Wazanaya Mandiveni from CBD, and she'll be speaking on issues related to what next, looking into the future under COP-MOP 11, issues on synthetic biology. So she will be presenting that. Wazanaya actually is leading the biosafety protocol in the CBD and has been in the landscape for quite a bit. She leads the biosafety unit, as I said, and also helps to coordinate the program, give guidance as needed. I'm trying to not to overspeak on the presenters. And in addition to that, she's been involved in the intersection of works on biodiversity and biotechnology, focused on balancing the need for innovation with environmental safety. So that is Wazanaya. The last presenter is Dr. Taemin Wu. If I pronounce your name wrongly, my apologies. Dr. Wu holds a PhD in Science and Technology Policy from KIPABiC, and her work focuses on biosafety regulation in emerging technologies, especially the political issues involved. And she's also been part of the Korean delegation to the COPs, focusing especially on biosafety and synthetic biology issues in terms of Korea's perspective. She also worked as a postdoctoral researcher at the Korean Biosafety Clearinghouse, where her interests were in policy and regulatory research. So that will be the last speaker for the day. I now call on Miss Wazanaya Mandiveni. Sometimes when you know colleagues, you don't mention their full name, so my apologies. Over to you, Wazi.
Thank you very much, Alex, and good time of the day to you all. And thank you for affording us as the Secretariat an opportunity to participate in this webinar. It's been very interesting to hear the perspective, and even for myself to get some of the history, since I've only been with the Secretariat and this topic for a short period of time. So thank you very much to both Felix and Felicity for their input. I'm not sure if I'm sharing the presentation myself. Can I?
Yes, please.
We were asked to look ahead to COP-MOP, to COP and MOP 16, to COP-MOP 11 and COP 16, what is coming up next for synthetic biology. And I have to say that I do not have a crystal ball, and so I'm not able to share what will actually happen next, as Felicity indicated. There are a lot of outstanding issues, and we are waiting to see how these issues will be resolved. And there have been, as we were told, a decade of discussions on synthetic biology. And in all of these processes and discussions, a number of structures have been created, as Felicity indicated. We have 5 previous decisions, 3 ad hoc technical expert groups, and 1 multidisciplinary ad hoc technical group that have been created specifically on this topic. And in these decisions that have been taken, a number of elements have been established, and COP16 will build on these previous decisions and highlight several aspects of the work that has been done to date. So the early decisions relate to including elements such as the precautionary approach for LMOs for biofuel production, as well as synthetic life cells or genomes. This was in the earlier decisions. By the time we got to COP13, the operational definition proposed by the ATEC was acknowledged, and this operational definition remains in effect. And you will see in all documents, publications, and outcomes, this operational definition continues to be the basis of the discussions. Decision 1419 recognized the need for horizon scanning, as well as the precautionary approach for LMOs containing engineered gene drives. And then decision 1531 established a process for horizon scanning. And it also decided not to require further analysis on new and emerging issues. There is, however, continued debate amongst the parties, as we saw at SABSA, as to whether or not this discussion is actually completed or not. We have had 2 technical series publications, the first one in 2015, technical series number 82, and the second, which Felix spoke about, where he's a co-author, in 2022. A key element that continues to prevail is the coordinated approach, and this is then between the convention and the protocol. And we have tried to maintain this coordinated approach, particularly within the program of work, by trying to always ensure there's an interface between both processes and trying to make sure that we have members represented from both processes for that consistency. And that's why we have members of the process, such as Felicity, who takes part in both the synthetic biology process, as well as the risk assessment process under the protocol. I won't speak more about this, except to say that decision 1531 established a process, and this process had 4 key steps: information gathering, compilation, organization and synthesis, assessment, and reporting on outcomes. And the role players for each of these processes was established. And this is the process that we've just finished, which Felicity spoke of, so I will skip this. And just stop here a little and share with you where we are and why we think COP16's work is well placed. Firstly, the first horizon scan monitoring and assessment process has been concluded. And this document is available, and COP will have this document before them, and we will have COP16 accepting and recognizing that the first horizon scan, which was asked for by COP-1531, has been completed. And this horizon scan identifies 5 key trends and issues, as well as another set of 17, and then altogether 54. So 54 trends and issues have been identified, and this horizon scan for synthetic biology now exists, and we hope COP will endorse it. The first multidisciplinary ATIC has also been established, and along the way, we've also learned a lot about multidisciplinary ATIC, what makes this one different, how is it different, and what have we learned about multidisciplinary. So this has also been done, and this is something which the COP will have before it. We have 35 options for capacity building, technology transfer, and knowledge sharing. Again, this is an important development which is before the COP, and we are proud that the process has come this far. We have a refined methodology for conducting horizon scanning. So now we know how to do it. And Decision 1531, a process was established, but the methodology was not clear. We now have a refined methodology, and if we are going to go forward, COP now has much more clarity on how this horizon scan could happen in the future. We've also done a literature review and learned some lessons along the way on issues of synthetic biology. It's complementary and it's a quantitative analysis. Again, before we went to COP-15, we didn't know how this could be done. We've now learned how to do it, put in place the infrastructure to do it, and we'll be able going forward to have this as a data set that's available to the parties. And the outcomes of the process were peer reviewed, and this information is available. So this is the basis for the work ahead for COP-16. In Nairobi, during Substa 26, a number of discussions were held, and we wanted to just highlight this because when you look at other topics, this was one of the most resourced topics to say, where we had a number of statements received. 45 statements were received. We received a number of inputs that really are reflected in the diversity of views. Our contact group met twice in an attempt to find common ground. And while there are many, many, many brackets present, there are also a number of options and a lot of areas of further development that COP16 can actually look at, because what Substa has provided to COP16 is a very detailed path, which many options available, and COP will then resolve them. So while there are many brackets in the Substa recommendation, the brackets actually represent options and opportunities. So the substantive elements from the Substa recommendation, and therefore the starting point for COP16, relate to a number of elements. Most importantly, the thematic action plan for capacity building, development, transfer, and access to technology and knowledge sharing. And from the discussion in Substa, it was quite clear that this thematic action plan is something that had broad support in the room and actually responds to one of the findings of the horizon scanning around inequity. So this also links well to the work of the long-term strategic framework for capacity building for the Kunming-Montreal Global Biodiversity Framework. So we see that the work on the thematic action plan for capacity building is a good element for further development. Within it, most of the text is relatively clean and agreed. However, there is a conceptual element that needs to be addressed, and this relates to whether it's a thematic action plan for synthetic biology, including biotechnology, or it's a synthetic, or its focus is on biotechnology, taking into account synthetic biology. Then the next issue, which is before the parties in the recommendation, is the future process on horizon scanning for synthetic biology. This is related to some of the elements which were identified in the horizon scan. So what should happen with integration of artificial intelligence and machine learning, LMOs containing engineered gene drives, and self-spreading vaccines for wildlife. So these are recommendations from the MATEC itself, and how this work will go forward is one of the elements still outstanding. The next outstanding area relates to a number of smaller aspects that are integral to the overall process. So some parties are again raising the issue of the operational definition of synthetic biology, that perhaps after having left this item alone for a while, this is now the time for parties to reconsider and look at the operational definition, and perhaps the MATEC can look at this now. There are some parties that still would like to clarify the new and emerging issues element. Again, this is text that is in brackets at the moment and an issue that parties would like further reflections on. Then the next element is the relation between applications of synthetic biology and the Cartagena Protocol. This is an element which is constantly under discussion, and we will expect further clarity in this regard, bearing in mind always the coordinated approach, as well as the fact that the Cartagena Protocol scope is really about transboundary movement, whereas the convention and the approaches for synthetic biology are a little bit wider and look at the potential positive and negative effects, and have greater elements that go beyond the risk assessment and biosafety aspects. So these are areas where parties are still debating, still looking for clarity, and we hope that COP16 will take a decision that takes some of these discussions forward. The next area is the recognition of the need to address inequity in the participation of developing countries in the field of synthetic biology. And this was, again, one of the trends. And we are still looking at how this then gets addressed in light of all of the other broader discussions that happen under the convention itself. Finally, the processes will then need to be further defined. We have learned how to do these things. It was a first attempt, and lessons have been learned, and a review has been undertaken, and we now need to look at how to put all of this together. And so the annex to the decision carries a number of elements outstanding on how will we proceed with horizon scanning. In addition, linked to this topic, COP-MOP 11 to the Cartagena Protocol on Biosafety will have a discussion on risk assessment, And this discussion on risk assessment, which includes the additional voluntary guidance materials to support case-by-case risk assessment of living modified organisms containing engineered gene drives. You see that the engineered gene drives are part of the outcomes of the horizon scanning process. And the MATEC also made another recommendation to the Cartagena Protocol on development of additional voluntary guidance materials for self-limiting insects. And this will be a discussion that will be considered as well, which is relevant to the topic of synthetic biology. And under agenda item 5 on compliance, parties will be asked to consider the implications of diverging interpretations of the definition of living modified organisms under the Cartagena Protocol and the varying legislative approaches among parties concerning new developments in biotechnology, including genome editing. And we see this discussion under the compliance as also relevant to the discussions on synthetic biology. We look forward to seeing many of you in Cali, and thank you for your attention.
Thank you very much, Wazi, for the presentation. The presentation has laid a roadmap on our preparatory processes for COP-MOP 11 and COP-16. I would pause with not make further comments, but we'll address the questions later on. But some of the key issues that she has raised, which we need to look at, is to go back and read the decision in 1531, look at the trends recommended and options for capacity building. And let's prepare to be able to come up with a compromise approach with hopefully removing all the brackets so that we can clearly have a situation where we have a global direction and also national perspectives to guide the way forward. Thank you very much, Wazi. We will come back to the questions at the end of the presentations. We will now look at regional perspectives, and I will call on my colleague, Dr. Viva Ahuja, to look at Asian insights on the complexities of synthetic biology regulation. She's already been introduced, so I will not attempt to introduce her again. Viva, over to you.
Thank you, Felix, for giving me this opportunity to share a brief perspective from the Asian region. Having been worked in this area, I would just like to share very briefly on a little bit of the kind of priorities which I feel that the Asian countries, in general, need for this, the benefits of the synthetic biology and also the regulatory part. So before I move on to the key issue, I would like to just give a very brief glimpse of what is the status of biotechnology and biosafety in Asian region. See, if we look at Asia, it is the largest and the most populated continent of the world. It's very, very diverse, economically, socially, politically, environmentally. 60% of the world population is here, 4.81 billion people. And as I said, there's a high disparity in economic development, natural resources, and most importantly, experiences in adoption of biotechnology. So synthetic biology is like, the much more advanced version, but we have had a very different scenarios with respect to modern biotechnology itself. And then we have a very varied, status in biosafety regulation across the region. In spite of that, there are yet some commonalities as most countries face a lot of challenges, particularly with respect to the healthcare and agriculture. Sorry, it's not moving. Yeah. If you look at the key challenges, as I said, we are highly populated Almost 351 million people are undernourished in Asia and Pacific region, which is more than half of the global total. High burden of infectious and non-communicable diseases, and an urgent need for affordable health care. At the same time, we have all these new issues like depleting fossil fuels, rapid environmental deterioration, climate change. I just put a small figure here from an FAO report which says the scale of agriculture, the impact of on agriculture of the climate change is going to be maximum in Asian subcontinent. So now if we look at the biosafety regulatory framework, most countries in Asia are signatories to the Cartagena Protocol on biosafety. And these frameworks have been in place mostly not because of extensive research and development in the product pipeline, but as a result of support as a party to CPB. So we have competent authorities designated in most countries. There were not separate laws enacted, but instead in the existing laws itself, the policies and the rules, etc., have been added. And biosafety regulations so far have been more inclined towards plants. There's very limited experience with most other categories of organisms in most countries. Like if you look at the examples, these are the countries which have adopted GM crops as an example, and we can see that the numbers are very small, and also the number of crops are much lesser as compared to the global scenario. Similarly, if we look at the upcoming technologies like gene-edited crops, there have been some headways. At least 4 countries came out with the clear regulations in Asia and Oceania. In fact, Australia is not to be included here, but since I took this picture from ISA, it includes Australia also, and all other countries where there is some noticeable policy-making discussions going on. However, there are, slow decision-making. Now coming to, some of the areas, a large number of areas which were highlighted by Felix in terms of the potential of synthetic biology for, the benefits part, as Felicity highlighted. I was just trying to look upon some summary, relevant areas if they have been summarized, and I came across one reference where 3 areas have been identified, which I think reflect the needs also in terms of next generation biomanufacturing, where you have energy crops, agriculture crops, novel functional materials, specialty chemicals, biofuel production by algae, future medicine. There are lots of areas, and then food, agriculture, and environment. Now, just a glimpse of this itself shows, just a quick look itself, how relevant they are for Asia. And we really need to work on, making use of these novel technologies for the benefit of the Asian countries here. In fact, many countries are now coming up with policies for this kind of, what we call bioeconomy, biomanufacturing, and so on and so forth. I would like to share here that just about a month back, or maybe less than that, in India also, our Prime Minister has approved a new policy which has come, Bio E3 policy for, bioeconomy, setting up biofoundries and so on, and all that would include, the synthetic biology applications. So how do we harness the benefits of synthetic biology in Asia? And we need to really focus on harnessing these benefits because the challenges are huge. So if we look at this, then encouraging innovation through trainings, through infrastructure, funding, all that is required in especially large number of the Asian countries. Regional collaborations are also required for capacity building, and so that, whatever they do also is as per the global standards. Of course, there are public biosafety concerns with every new technology, the way we have seen genetic modification and then gene editing, also some concerns, although there has been a much positive, it has been much positively received. And then integration within the existing laws. We have seen that despite, 2003 to now 24, more than 20 years of Cartagena Protocol, there are many countries where still new laws are stuck. The biosafety frameworks are not very highly functional and so on. So creating new and new guidelines and new regulations doesn't help. It only delays, bringing in the benefits of new technologies for the benefit of the society. Public engagement and awareness is always very important. And these are some of the key areas which we really need. And I would say in this context, the component in the proposed decision, proposed recommendations by SBSTA on capacity building and development, access to and transfer of technology, and knowledge sharing in the context of synthetic biology is extremely important for the Asian region. These are the few points. I have only picked up this because a lot has been said about horizon scanning by Felicity also, and then by Wardzanai. But from my point of view, I think what is in, what is out is not, will continuously be discussed, and it will be this one. But we need to really focus on capacity building and parties and stakeholders. Whatever these are some of the points. Emphasis on the capacity building and development of the technology transfer initiatives, parties and other stakeholders to facilitate broad international cooperation, knowledge sharing, capacity building, and then thematic action plan to support the above. Of course, the issue is that whether it should be biotechnology or it should, because biotechnology includes synthetic biology. This is also one point of view. And then the suggested decision on these components is, of course, bracketed and hopefully will be resolved in the upcoming COP. So what are the key takeaways from whatever I wanted to say is that Asia is a complex landscape shaped by different regulatory approaches, diverse national approaches, different levels of innovation support. In many countries which are at very, Vardzanai also highlighted about inequities in the development and use of these technologies and evolving biosafety and biosecurity issues. However, it's very extremely important to note from the first presentation itself that applications of synthetic biology can really provide substantial benefits in the kind of challenges being dealt with, the challenges that the Asian region is facing. And so we really need to streamline these regulatory complexities, not going into too much of details, but in a very global and a streamlined manner, and make use that all available resources are used primarily for the capacity building and access to the technologies, so that the population at large gets benefited and has access to nutritious food in a sustainable manner, affordable healthcare, and so on and so forth. Thank you so much. This is what all I had to say. Thank you.
Thank you very much, Dr. Ahuja. Viba has raised quite relevant issues, which I hope you have questions too. But one of the fundamental take-homes that we will need to look at from the presentation so far is that this is simple and complex at the same time. Therefore, the parties, is very important that you involve national, regional, and global expertise in your dialogue to get a clear picture. Many times from over the years, when issues come, it is left either at the biosafety office or at the ministry. Meanwhile, there's a lot of national capacity. So hoping that within the region, you evolve that kind of core of people to look at these issues, knowing that the biosafety protocol, especially if this were to fall within, is a transboundary instrument. Therefore, if you build alone, you have not achieved much. Thank you. We'll go to the last presenter in this session, who is Dr. Wu. She is going to speak about the policy landscape in the Republic of Korea. Over to you, Dr. Wu.
Thank you, Alex, for your kind introduction. Okay. Hello, everyone. I'm Taemin from KAIST. Basically, I'm trained as a social scientist and studied science and technology policy. And I'm currently researching to map the social landscape of SynBio in Korea. I thank KIPABiC and Dr. Jang for this timely and meaningful event ahead of COP16. As Dr. Viva wonderfully introduced about the Asian region's perspective and mine will be a little bit more specific. I would like to introduce biosafety governance in South Korea and discuss the current policy landscape of SynBio. I believe that Korean case can illustrate, on the one hand, the political status of SynBio as a national strategic technology being pursued competitively in many countries, and on the other hand, the challenges that need to be overcome to achieve a sustainable environment and society. In Korea, the Social Climate for LMO Act was created in the 1990s with discussion at the CBD on adopting the Cartagena Protocol. Following adoption of the protocol in 2001 and implementing the LMO Act in 2008, Korea has developed a biosafety management system that includes a consultative review system with ministries responsible for each use of LMOs. This system is somewhat complex and fragmented, but is lubricated and sustained by institutions such as the Korea Biosafety Clearinghouse in facilitating inter-ministerial discussions and mediating consultation. Under this system, South Korea is one of the largest importers of LMOs for food and feed. More than 10 million tons have been imported every year. However, no LM crops are grown domestically. And the domestically developed GM grass went through the first risk assessment process for cultivation, was actually ultimately deemed unsuitable in 2023. Korea has had a mandatory GM labeling since 2001, and a new plan has been announced to phase in a full labeling of GMOs from 2026, subject to social consensus. To summarize, I think LMOs are everywhere in Korea, but also nowhere. The moments when LMOs become visible in Korean society are not routine, but quite dramatic, as they are often accompanied by controversies. In 1999, the greenhouses where GM crops were being tested by the Rural Development Administration became the scene of a movement calling for the safety regulations that were lacking at the time. And 15 years later, a GM sign stumbled upon by a farmer in Korea's grain fields sparked a nationwide anti-GM movement against the commercialization of GM crops, especially GM rice. In the spring of 2023, it was discovered that unapproved GM zucchini was being grown and produced in the country, raising concerns about national quarantine system and compensation measures. Ironically, apart from the prominent NGO activism at these moments, the level of social concerns about LMOs in Korean society remains actually very low. Currently, the most prominent anti-GMO organization in the country is Anti-GMO National Action, which was formed in the wake of the 2015 event, as mentioned earlier. And this organization has been campaigning against GMOs with an agenda condensed into these 5 promises here. This political and social landscape surrounding LMOs is essential for understanding the SynBio debate. In Korea, synthetic biology is a latecomer, but starting from the 3rd Biotech Promotion Plan in 2017, SynBio has received prominent policy attention and support as one of the 12 national strategic technologies as a key for realizing bioeconomy and biomanufacturing innovation. Korean government also recently has announced plans to focus on developing 9 core technologies of synthetic biology. However, whether the two goals of realizing biomanufacturing innovation and sustainability are on the same page will require rigorous review and continuous adjustment. In January of this year, Korea attracted worldwide attention again with its decision to support building a public biofoundry. As the national strategy for SynBio begins to take shape, regulatory discussions have also recently become more active. In 2022, an amendment was proposed to exempt novel biotech products, including gene editing technology, from regulations such as risk assessment through prior review if no foreign genes are introduced or remain. In 2023, a special law to promote SynBio was proposed, and discussions are underway to create a more relaxed regulatory map for SynBio R&D and products. Unfortunately, discussions in the National Assembly did not progress much, and both bills were abandoned due to the expiration of the National Assembly's term in last May. However, discussions are likely to continue as bills have been recently reintroduced. The most prominent issue in the debate on the regulation of SynBio in Korea is, I think, the definition of synthetic biology. Current position of the Biosafety Management Organization is that organisms derived from SynBio are covered by the LMO Act. However, there is a contrasted view in the research community that non-living organisms, or specifically artificial cells, that are synthesized from scratch without using existing organisms fall outside the scope of the LMO Act. While there is no agreed legal definition in Korea, recent policy documents have used definitions as noted here, highlighting specific examples such as DNA, proteins, and artificial cells. Some experts have suggested that the LMO definition itself should be adjusted, and that a new definition for SynBio-derived products is needed, similar to the EU's creation of a new category of NGTs. As many of you have followed the process of creating an operational definition in the CBD will agree, it is important to avoid reducing the debate to a definitional issue, I think. It is also important to recognize that the discussion about the definition of LMOs is not simply a matter of a legal interpretation, but a matter of policy or even political decisions. Another issue is that there are calls for changes to the LMO Act in the context of the government's strong commitment to promoting SynBio, represented by BioFoundry these days. As the establishment of the BioFoundry will accelerate the volume and pace of SynBio experiments, there are calls for a simplification of regulations, especially for R&Ds and production using LM microorganisms. There are also continuous calls for the previously proposed amendments to the LMO Act, which include a prior review system for novel biotech products that do not contain or retain foreign genes to be reviewed in light of SynBio products. In addition, policy issues beyond the LMO Act have been also listed, but there needs to be more substantive discussion in Korea. Finally, I would like to summarize the last slides with these three keywords. The first one is absence. By exploring the policy landscape of synthetic biology, I found a lack of space for public engagement, as Dr. Viva also mentioned. There have been 2 technology assessments on SynBio and BioFoundry in 2022 and 23, but public awareness and understanding on SynBio are still very low, and there is a need to concretize different visions or promises of SynBio, recognizing that bioeconomy or sustainability, which are the foundation for the global rise of SynBio, can only be realized by considering a social alignment with much broader societal values. The second keyword is, I think, entrenchment. The debate on the regulation of SynBio in Korea is highly polarized mainly around how to bypass the LMO Act, or that it falls under the scope of the LMO Act, and therefore the current regulations are quite sufficient. Claire Morris, I would like to introduce, a British sociologist of science and technology, has proposed the concept of synbiophobia as one of the misconceptions of researchers about public scare of synbio. Based on this idea, the researchers' experience with the LMO Act and the negative public perception of LMOs in Korea seem to be creating a researchers' phobia of LMOs or LMO Act, and this phobia is particularly at work in the regulatory debate. In this circumstance, I think it will be necessary to create a space for diverse discussion in order to prevent the regulatory debate from stagnating and to open up the social evaluation of the promises of the field. The final keyword is translation. The debates that led to adopting the Cartagena Protocol back in the 1990s were a critical tipping point in the progress of the domestic debate in Korea. However, local impact of discussions in international treaty like CBD is not just a given, but requires an arduous and complex translation process. Since COP-15, multidisciplinary attack has been working to prioritize synthetic biology issues, but the implication of these issues in the domestic context have not been successfully contextualized, I think, and have not gained much attention in Korean society. Conversely, how to scale up the domestic symbiote discussion and contribute to international debate is also an urgent challenge we face ahead of COP16, which is just over a month away. I will end here, and thank you for your attention.
Thank you very much, Dr. Wu. I trust that the presenters will not mind that we share the presentations, because there are a lot of thought-provoking questions, some of which we will not be able to answer within the time space. Colleagues, for this session, we have looked at the global preparatory processes towards COP16 on synbio and related issues. We also had a regional perspective with some reflections for Asia, and then at least one country presentation on what is happening. The colleague from Korea has clearly highlighted the importance for national discourse impacting on global developments, the importance of practical hands-on attempts on addressing the issues, the importance of research and incorporating research to our policy discourse, among others. I will not attempt to summarize further. We have questions, and I also want to humbly ask the organizers, I may have to step out in the next 10 minutes because I'm leading another meeting. And if I do, without asking her permission, I'll ask Dr. Viva Ahuja to continue. All apologies, madam. So I'll pick a few of the questions that are here, which are still open. The first will be to my colleague Wazi and the other presenters. The question is, what will be the future of synthetic biology in 2050? It looks like people think you have a crystal ball. So Wazi, what will be the future of synthetic biology in 2050? The other two presenters can also add their views on it. It can be one question that can summarize a lot of the questions that are being asked. So over to you.
Thank you. Yeah, 2050. That is hard, but I can tell you that in the current process for our parties, we have just started to analyze the national targets that countries are submitting in their national biodiversity strategy and action plans. And in those strategies, what we are seeing is that parties are highlighting Target 17 as an area where they are struggling, and an area where particularly the biotechnology aspects of that target. So I think that we will still be having this debate about capacity and capability, because if the national targets for 2030, parties are struggling, 2050, unless something very radical happens, we will have the technology advancing for some and leaving others behind. And so I think that the emphasis should be on now, having these discussions and building on these discussions, and let's see what happens. So 2050, we will be where we are unless a concerted effort is taken to take everybody along with us. Thanks.
Thank you. Dr. Ahuja, any comments on that?
I think Wardzai put it nicely that the debates would continue, but I am very hopeful that there will definitely be more products coming out of synthetic biology, and hopefully these inequities would be removed to some extent, because the challenges are huge and we really need to work towards those challenges. So if, some debates will continue, of course, but I think more products and more, products and more technologies will come and will be useful for the Asian countries, particularly, that's what I look forward to. Hopeful of having more resolution and more products available for us. I am a biotechnologist and I really feel that research advances are just mind-boggling, and but at the same time, as she said, all these advances should need to be harnessed by everyone, by various countries, including the developing countries as well as the developed countries. The divide is big, but I'm sure there'll be more efforts to resolve that divide through the technology itself. So the debates, of course, will continue, but we will have, I see a more optimistic picture. Thank you.
Okay, thank you. I will just add the last question. Because of time, I believe the organizers would ensure that the panelists respond to all the other questions, and then we'll share with you in written format. This question summarizes about 3 or 4 of the questions that have been posed. It says, in what ways do synthetic biology-derived living products intersect with or diverge from the regulations and definitions established by the protocol? Basically, what are the regulatory concerns on synthetic biology, and how does it either merge what we know from the protocol or diverge? It's a very broad question. I would give all the 5 presenters a short time to respond.
Perhaps Felix should respond on the technical side.
Okay, good.
What has happened with the discussions under the Cartagena Protocol on Biosafety are really linked to the definition of a living modified organism. And that definition refers to a transgenic element. And so where you have a synthetic biology product that is not transgenic in nature, such as the genome editing, etc. This is where our parties are having differing interpretations around whether or not a product of synthetic biology would then be a living modified organism. And our experience to date is that the products that are out there as synthetic biology are living modified organisms and therefore falling within the scope of the definition in the Cartagena Protocol on biosafety. But there are some that are not. And as a result, that is where the difference is. And this is the discussion that will be happening. We hope not at COP-MOP 11. We hope that COP-MOP 11, the parties will agree to have a discussion about it, but not try and have the discussion at the COP-MOP itself. Thanks.
So in my view, the kind of horizon scanning has taken, the kind of areas which were listed so far, which was very nicely listed, and some 54 areas and so on. I would say most of these projects are going to be classified as LMOs, although they may need a slightly different risk assessment approaches, the way risk assessment for gene drives had to be, dealt with in a slightly different manner, although we're keeping the basic principles the same. But then, at the same time, as we are seeing that, the research advances are enormous. So it is very difficult to say whether all LMOs, all synthetic biology products will be LMOs. But as on date, I think most of them are the same. And so the regulatory approaches in terms of the coverage under the LMOs will be similar. However, slight deviations may be required based on the technology. Thank you. Alex, you are
Any
there?
other comment from the panelists? Yes, I'm giving the other people 5 minutes more. Any other comment from the panelists before I summarize my quick thoughts? Dr. Taemin, do you have any comment? Or Felicity, any of the two? Okay, so go ahead, please.
Yeah, not specifically, but even though most of the LMOs are LMOs fall within the scope of LMO definition of the Cartagena Protocol, that doesn't mean that there's nothing to do, nothing we can do. Like there will be more products that aims to release to the environment. So I think there is many things to do at the CBD to discuss about how to deal with these new types of organisms, even though they are fall within the scope of LMOs. That's my final note.
Thank you very much. Felicity?
Yes, for me, I would agree. I think WOTSI and VIBO have covered the conclusions from the programs of work so far, in that most LMOs developed or currently in development are within the definition of LMO, and that's most organisms. And there's some divergence of views, as WOTSI mentioned, on whether or not all organisms developed with the use of genome editing fall within the scope of that, and that's where there is some parties are of the view that they should fall within it, and there's others that there are many countries now that have clarified status of those organisms at national levels, and that includes that some of these are not LMOs. So at the international level, there is an agreement on that. There was also some discussion in the RTEGs around, there was some of the products, there was discussion about whether or not they were living. There are some entities developed, if it's not living, it's not a living modified organism. So there's other types of products developed here that don't fall within the definition because they are not living modified organisms.
Okay. Okay, thank you. So thank you very much. I was getting a message. Are you joining the other meeting? We've had a very interesting day. Before I hand over to Dr. Jang, I think what we've done is to generate further discussions amongst the parties and other stakeholders involved in this process. Going to the COP is very important that we go over the program, the material developed so far, and develop clear issues that we would want to see going through a decision, and also as much as possible dealing with the issues related or highlighted in the brackets. I also want to emphasize that we will need the challenge, having worked in government before and in the region, the challenge we face is that sometimes the legally competent authorities shy away from expertise from academia, from research, society, private sector, and the rest. It is very important that some of the issues that we are discussing might even be happening in the country. So the focal points and the competent authorities will have to engage, because after all, the obligation is to the parties, that is the whole of the country, and not just the ministry. So it's a point that I want to highlight. I also want to pose this to those parties that go to the COP-MOP. Is it time to look at Article 29 para 4E, which allows parties to review progress and possibly even amend the protocol or develop new annexes, which can help them to implement the protocol in a more sustainable manner? Are we yet at the point of amendments or development of new annexes? At the last meeting in Nairobi, some parties were also suggesting that because SynBio is both convention and protocol related, are we willing to have a new protocol on synthetic biology? I leave these questions for us to ponder over as we prepare to go to Colombia. Thank you very much, and over to Dr. Jang. Thank you, and have a good weekend for those who are still on Friday and also Saturday. Thank you.
Thank you, Alex. Well, originally we planned a 100 minutes webinar, but I see more than 2 hours passing. But I think it's worth it. Yeah. And for which I thank you all, especially all that together to the end. And I want to extend a heartfelt thanks to each and every one of you who made this event complete and successful. A special thanks to our esteemed moderators, Dr. Alex Wusbini and Dr. Viba, for their expert guidance and for leading us through the sessions. I would like the same to go to our distinguished speakers, extending my thanks for sharing your knowledge and experiences with us. Your insights have enriched our knowledge and discussions. I also wanted to express our gratitude to all participants who joined us from around the world. Your active engagement and thoughtful questions have greatly enhanced the quality of our discussions. I understand many of your questions could not be answered. Like Alex mentioned, we'd like your answers be left in Q&A in written form. then the experts that was present here may pitch in, or other experts could help with this. And we hope that the knowledge gained today will be beneficial to you in the varied roles that you play. Finally, thank you to the organizing team at KIPABiC and our partners at UNEP, BCIL, and the participating countries. As we look forward to continuing these important conversations and collaborations, we invite you to take a moment to share your feedback on today's webinar. Please also let us know of any future areas of work you would like us to pursue. Your input is crucial in helping us tailor our future initiatives to better meet your needs. In this context, I will give you a small announcement. Well, like Dr. Viba mentioned, to further continue these discussions, we will be hosting a webinar on risk assessment management ahead of the COP-MOP meetings later towards the end of this month. We will share details soon. Thank you once again for your time and participation. Thank you.